Hey guys, welcome back. Last time we talked about pedigrees, we talked about a few different ways we could track inheritance. Now we're going to talk about some chromosome changes. On rare occasions, a chromosome structure can actually change. That's generally not a good thing. They're usually harmful or lethal. Very rarely can a chromosome change its structure and have this be neutral or even beneficial. Transposable elements are one of the big workhorses for changing the structure of a chromosome. They alter chromosome structure because they move. They're sometimes jokingly called jumping genes, like jumping beans, and they move spontaneously within a chromosome or even between chromosomes. They can just break themselves off and jump over to a completely different chromosome. And sometimes that can be not a big deal, and sometimes that could be like a really, really, really bad thing. So a segment of a chromosome also can be duplicated or deleted or inverted or translocated. Duplicated means you make a second copy of it. Deleted means you remove it. Inverted means you break it off, turn it the opposite direction, and reinsert it. And translocated is you break it off, move it somewhere else, and reinsert it. And these are pretty much all going to be really bad things. So here's duplication. The section of the chromosome gets duplicated. And so here you have this band of genes and this band of genes. And for whatever reason, when it went through replication, it made a second set of those same genes. The chromosome is much longer now, and it has this duplicated set. And some species are pretty tolerant of those kind of changes, but humans tend to not be. Deletion, here we have a chromosome with this big old gene set in it. For whatever reason, it was broken off and lost. The chromosome is much shorter, and that gene is completely gone. Bad thing. Here's an inversion. I wish they would have made these different colors. That's okay. A section of the chromosome gets flipped so that it runs in the opposite direction. So we've actually broken this off, and flipped it around. And now the top is the bottom is the top. So I actually grabbed this and rotated it, inserted it upside down. Bad, bad deal. And then finally the translocation, not the same as the beneficial crossing over you learned about in meiosis. This is a broken chromosome gets reattached in the wrong place. And here we have two non-homologous chromosomes exchanging pieces. This is a different chromosome, maybe chromosome, I don't know, 20. Here's chromosome 18, and a leg of 20 goes over to 18, and a leg of 18 goes over to 20. Now these chromosomes, they're not even homologous anymore, and this animal or person or plant is probably going to be in a world of hurt. So that's translocation. So, chromosome change is supposed to be one of these things that's kind of driving evolution, and I don't know. I think it's mostly a bad thing, but I'll let you be the judge. Change in the chromosome structure can reduce fertility in your heterozygotes, so less likely to produce offspring. However, the accumulation of multiple changes in homozygotes could potentially give us a new species. Uh, your call. Um, Changes might make you infertile, but if we get enough changes, then maybe we can become a completely different species. Sure. Certain duplications might allow one copy of a gene to mutate while the other carries out its original function, sort of as a way of maybe trying out new strategies without completely committing to them. The example is they believe that the X and Y chromosomes were at one time, long, long ago, homologous autosomes in reptiles, not just reptiles, reptile-like ancestors of today's modern mammals. They thought that those reptiles, like today's reptiles, their gender might have been dictated by the weather or the climate that they were hatched in, and that one of these Y chromosomes, the X, lost a piece and became the Y, and then that made up males, and then in the future, everything that's evolved out of the reptile-like ancestors of mammals, now all of the mammals that run down this evolutionary branch make distinct male and females. Right, there you go. So here's the idea. Ancestral reptiles, pair of autosomes, which are body chromosomes, 350 million years ago maybe, sex being determined by temperature. SRY gene begins to evolve, and the DNA sequence of the chromosomes diverge as we get more and more mutations in what we're now denoting as the Y chromosome. 320, 240 million years ago, DNA sequence of the chromosomes are so different that they can't cross over in one region because of all these mutations. The Y chromosome becomes very short. 3 more times, the pair stops crossing over again and again and again over, just like 100 million years. Each time the DNA sequence of the chromosomes diverge, the Y chromosome shortens and shortens and shortens. Today, the pair crosses over only in a small region near the ends. So this is the human Y chromosome now, which determines our gender. I wonder if it's still shortening. Maybe it's still shortening. I wonder what my kiddos' genders will be determined by 100 million years from now. Anyways, there's the idea. Take it for what it's worth. Tiny Y chromosome. Miraculous. Let's look at some differences among some more closely related organisms. Here we have humans, 23 pairs of chromosomes. Chimpanzees, gorillas, orangutans, they have 24 pairs of chromosomes. One of the ideas here is that 2 chromosomes potentially could have fused end to end at some point during the idea of human evolution. And so the argument is that here's one of these human chromosomes, and here's a couple different chimpanzee chromosomes. And so the idea is like, well, how do we get from 24 chromosomes in chimps, gorillas, and orangutans and 23 in humans? Well, what if we took these 2 chromosomes that look similar with genes that look similar? What if they overlapped and attached to each other here? We would get something that kind of looks similar to what's in humans today. So maybe that happened a long, long, long time ago and is what branched humans. Wow, should I say that? What branched what was going to become modern day humans from the branch that was what was going to become modern day gorillas, chimps, and orangutans. Cool. I don't know. It's an idea. There you go. Chew it over. Heritable changes in chromosome number. Sometimes these abnormal events occur before or during meiosis, and new individuals end up with the wrong chromosome number. Consequences range from minor deals to lethal changes. 2 examples of this are polyploidy and aneuploidy. A lot of flowering plant species, a few of our insects, fish, are polyploid, which means they have 3 or more complete sets of chromosomes. We've said that humans have 23 pairs, so we have 46 total. Polyploid would be like, instead of 23 pairs, 46 total, you'd have to add another 23 to that, and another 23 to that, and you become 3N, and 4N, 5N. You don't add 1 chromosome, you add a whole additional set of 23 chromosomes. That's how you become polyploid. Humans do not tolerate that, not at all. A lot of plants can, but boy, humans, bad news for us. We can barely tolerate aneuploidy without just dying right away. Sometimes you see trisomy and monosomy in aneuploidy, which in individual cells have too many or too few copies of a chromosome. So trisomy is you should have 2 pairs of a chromosome and you have 3 pairs of a chromosome. Somebody got hooked up during meiosis or broke away during meiosis that shouldn't have, and instead of having 2 sets, you have 3 sets. So instead of having 46 chromosomes as a human, you'd have 47. Or instead of having 46, you would have 45. And if you're lucky, you can survive depending on which chromosome this happened to. But we're not very tolerant of those changes. So one of the reasons you get these is what's called non-disjunction. To disjunct is like to break apart, so the not breaking aparting. Changes in your chromosome number can be caused by non-disjunction. A pair of chromosomes fails to separate during mitosis or meiosis, and so instead of one sister chromatid being drugged together into the cell, both are drugged together into the cell, and now our chromosome number is all messed up. So it affects the chromosome number at fertilization. If you're monosome, you have n minus 1, like I had mentioned before, 45. So if this is a gamete, instead of 23, it would be 22 chromosomes in an egg or a sperm. Not a good deal. If it's trisomy, it's n plus 1, so 23 pairs would be 24 chromosomes in an egg or a sperm. Not a good deal. Not something humans are going to tolerate well. And so here it is. Metaphase 1, we got a pair, everything's cool. Anaphase 1, we got a pair, we got 2 pairs. Not everything is cool anymore. We're the opposite of cool. Spindle fiber is busted. Both chromosome pairs are being drugged to this side, and now everything's all messed up. So we're going to go ahead and make, I don't know, sperm, eggs, pollen, whatever. One set's over here. That set gets split. Not cool. We have one set. These sets are over here. They get split. Not cool. We have 3 sets. Our goal is 2 and 2. We got 3, 3, 1, and 1. All these sperm, all these egg, they're all messed up now. And if they're fertilized, it's tough to say if that thing's going to survive or not. So there's metaphase 1, anaphase, okay, yep, we got it, okay. So our most common example of this in the United States by far is Down syndrome. Only trisomy 21, to our knowledge, allows survival to adulthood. All these disorders are either going to result in death immediately, like right after conception, or they're not going to make it until they're an adult. They're going to die real young. Only trisomy 21 can survive into adulthood. They can live pretty normal lives, but I mean, you guys kind of know how this goes. Physical appearance is a little bit different. Mental impairment, heart defects as they age, all sorts of different things that you get with trisomy 21. The incidence of non-disjunction increases with maternal age. If the mother is 25 as opposed to 35, as opposed to 45, the older the mom is, the higher the risk of non-disjunction occurring. And I think there's a graph in your book on this. If there's not, ask me in class and I'll pull you one up on Google, because the rate that risk increases is just about mind-blowing, and it's interesting to talk about. It's a fun talk. Sad talk? Fun talk? I don't know. We should have this talk in class. This can be detected through prenatal diagnosis, but whether you want that information and what you would do with it, that's up to you. And so here's your karyotype. The chromosomes are sorted by number and by size. So we have chromosome 1, 2 pairs, chromosome 2, 2 pairs, chromosome 3, 2 pairs. We're good, we're good, we're good, we're good, we're good, all the way down. Chromosome 20, 2 pairs, chromosome 21, 1, 2, 3. Trisomy 21. Chromosome 22, we're good. X, we're good. Y, we're good. We have a male with Down syndrome. So when you look at the size of these chromosomes, just kind of off the top of your head, if you had to guess, if we get a trisomy in anything else, it results in death, like right away. Look at 21. Why do you think that a human can tolerate having 3 copies of 21 without dying right away? Hopefully you noted the size. It's about the shortest chromosome we have. There's not that much information on it. Well, there's a lot, but compared to one of these things like chromosome 3, it just goes on for days, you know. So yeah, it's a really, really, really short chromosome. Here's your book, has this this comment about the eyes, specifically the irises of children with trisomy 21. One of the things in the phenotype that you'll see a variety of things, shorter fingers, smaller hands, things of that nature. But one of the phenotype giveaways for trisomy 21 is having a starburst iris. See this eyes kind of this brown hazel color, and in it are yellow starburst or yellow dots with stripes running to the inside. And I'm not sure why that is, but it's just one of the things that is expressed in the phenotype of a child with or an adult with trisomy 21. So kind of fascinating. So what if we change the sex chromosome number? You wouldn't think this happens that often, but it actually kind of does. Um, it generally impairs a lot of things other than just the sex chromosome, learning, motor skills, those sorts of things. And sometimes these can actually go undetected for most of the life of a person. Female sex chromosome abnormalities, they can have Turner syndrome, which is XO, so they inherited an X chromosome from, mom or dad, somebody, and they inherited nothing from the other parent. So they only have one X chromosome, and so you get a female, of course. It can't be a male. The other option is XXX syndrome, where they have three, or sometimes more, X chromosomes. Of course, it's a female, a super female, you could almost say, because there's so many X chromosomes, but really is a little bit of mental impairment, things of that nature. So I wouldn't say it's necessarily a good thing, but they can survive and even produce offspring, so there you go. There are some male sex chromosome abnormalities. Klinefelter's is a male who has an X chromosome, an X chromosome, and a Y chromosome. And then there's also XYY syndrome, where you have a male who has two Y chromosomes. So there's pretty much every variety you can think of for abnormalities in sex chromosomes. And a lot of these guys or gals, you'd pass them on the street and not even know it. Really, you wouldn't even know they had this. So those are some chromosome abnormalities. All right, so let's go ahead and stop there. We'll pick up genetic screening and these last couple sections in our next video. So thanks for your time.